Hi everybody, welcome back. Today we're going to be doing bacteria. This is lab 11, page 79 in the lab manual. So the objectives for today talk about background on bacteria, shapes and characteristics, good and bad bacteria, how to grow microorganisms, including bacteria, talk about skin bacteria specifically, upper respiratory bacteria, and then we'll talk about your homework for next week. And just a reminder, next week is technically Easter break, so you don't have anything due on April 9th or 10th. Everything will be due on either the 16th or the 17th. Again, check with your Canvas professor specifically to know when things are due. So starting on page 79, some background on bacteria. Bacteria are old. They are some of the oldest organisms on the planet, even 3.5 billion years older than us as humans. They also live everywhere. Soil, your bacteria, your gut, your skin, in the hottest places on earth, in the coldest places. They live everywhere. They also have so much of a bad reputation. We give them a bad rep. We treat them bad. We try to clean and, this Lysol is disinfecting, or we have a lot of soap. We don't want children to get sick. But there's something called the hygiene hypothesis, which states that having an early childhood exposure to bacteria, such as in soil, can cause stronger immune systems and decrease incidences of allergies or asthma. So having an early exposure builds up our immune system. Have you heard of rub a little dirt in it, or just go play outside and you'll be fine, eat some dirt? It's kind of that idea. Bacteria come in multiple shapes, and the ones we'll be talking about are coccus, bacillus, and spirillum. Coccus are round, and the way I remember it is coccus has an O, and it's like a circle. The plural is cocci. If you've heard of like streptococcus, staphylococcus, that's this. It has its shape in the name. It's a descriptor. Bacillus is rod-shaped, so here it kind of looks like a little rod or kind of like a long pill, it's an ovally type shape, and that bacillus can also be in names of different bacteria. And then spirillum, it's in its name, it's spiral shaped. So here they're kind of squiggly. They can look somewhat like bacillus, like this, just a little, not as long, but generally they tend to be squiggly shaped. We also characterize bacteria by their makeup. So, for example, on page 80, there's cell wall. It talks about the Gram stain in a peptidoglycan cell wall. Peptidoglycan is a protein that makes it really a thick wall. And we use a Gram stain, which I won't get into too much, to determine if it's Gram positive or Gram negative. And here we have different colors. This is more of a purple, and these two are more pink. Gram positive has the peptidoglycan. It keeps that stain in there, so it's Gram positive. And these two do not have peptidoglycan. They are Gram negative, so they are more of a pink color. Bacteria are also prokaryotes. So prokaryotes means that the bacteria cell does not have a nucleus or organelles. You, as a human, are a eukaryote, which means you have a nucleus and organelles in all your cells. Bacteria are single-celled and they are prokaryotic. We also can break down how they make food or eat food, being heterotrophic or autotrophic. Hetero means different, whereas auto means one or self. So you can think about it as a heterotroph has to go somewhere different than itself to eat. So you go and you find food. Usually bacteria who are heterotrophic feed on dead organic material. Autotrophic means you make your own food. So you, oneself, make your own food. That can be photosynthetic, like plants, so they make food from getting energy from sunlight, or they can get their energy from inorganic molecules like hydrogen sulfide. They also reproduce asexually, so the organisms that they have, the offspring that they have, are genetically identical to themselves. We can also define bacteria by how they, either if they use oxygen or don't use oxygen. So obligate aerobes need oxygen. So you can think about it, aerobe has the word air in it. They need oxygen. And obligate, I'm obligated to have oxygen to live. Facultative anaerobes, so here we have anaerobes, so it's anaerobes, no air, Facultative anaerobes can use oxygen for respiration to make energy, or they switch to fermentation where they do not use oxygen. Very similar to how we make wine or ferment like pickles and things like that. It's without oxygen in it. So they have a choice. They choose between. And then we have obligate anaerobes where they cannot have oxygen. They are killed by oxygen. So going to page 80 and 81, good versus bad bacteria. Bacteria, like we said, has a bad rap, and we generally view it as bad, but it has a lot of good things, and I like to call it cheeses and diseases. It has good things and bad things. So certain bacteria can fix nitrogen. 79% of the air around us is nitrogen, but it's in an unusable form, and so there's bacteria in the soil that actually helps plants fix nitrogen and convert it into a form of nitrogen that we can actually use, which is usually ammonia. And then we end up eating the plants and get the nitrogen from there. We need nitrogen. We just can't use the kind that's in the air. So bacteria helps us fix nitrogen and use it. There's photosynthetic bacteria that produce oxygen. So just like how plants take in carbon dioxide and give off oxygen, photosynthetic bacteria do the same thing. So they put oxygen back into the environment. Bacteria also make food, antibiotics, and insulin, so like cheese, sauerkraut, yogurt. They also make antibiotics and insulin and other things like that. A symbiotic relationship is a relationship where two things are giving each other things that are helpful to the other, kind of a, not a give and take, but in the example of your gut bacteria, they are feeding off of nutrients in your gut, and you are also getting vitamins and protecting us from pathogens. So we're both gaining something from having the bacteria in our gut. Bacteria are mainly decomposers, breaking down dead things, just like how fungi do it in the forest and all over. Without bacteria and fungi, we would have so many dead things everywhere. So it really is helpful that it breaks down, and it provides new life by converting it into organic matter. Now, I will talk a little bit about how bacteria can be pathogenic. That means they cause diseases. Just like how we talked about the hygiene hypothesis, having exposure to common bacteria can help build you up so that your immune system knows what to respond to and what to ignore. But that does not mean that poor sanitation is a good idea. We shouldn't go and lick doorknobs, especially not right now, just to expose ourselves to toxins and bacteria. That is not a good idea, especially right now. Another thing to be wary of is antibiotic resistance. So oftentimes physicians and health professionals prescribe antibiotics as a way to get rid of bacteria, even if it's not totally necessary. For example, I had an ear infection. I thought my ear had some buildup as well, and they were like, well, we can't tell if it has an ear infection, but we'll give you antibiotics anyway. Having bacteria exposed to multiple antibiotics over time can lead to antibiotic resistance, and then you can have bacteria that are resistant to multiple types of antibiotics, and then you have almost what we call a superbug, so that it can't actually be killed by any antibiotics. So it's really important that you don't overtake antibiotics. Don't just take them unless you're prescribed them, and even maybe question your doctor if they prescribe them. And another thing is also take the full course of prescribed antibiotics. Don't just take it for a few days. You're going to feel immediately better when you take antibiotics, but make sure you're taking the full dose so that the bacteria are totally killed and not have an exposure to it, a weakened dose. So bacteria can be good or bad. How do we grow bacteria in order to learn more about it? This is on page 82 in the lab manual. So bacteria are very small. They're very, 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 very tiny. A single bacterium, you can't even really view it with your own eye. And even the pictures I showed on the earlier slides, we are zooming in over 1,000 times normally on a microscope for you to see that. We grow them on petri dishes that have agar in them. Agar you can think of as basically a gelatin that forms this nice flat surface for them to grow on, and then we have food inside that they can feed off of, and so it grows certain bacteria on the plate. They grow in these little colonies, so these little circles, and it looks like just one circle to us, but when you zoom in, it's actually tons and tons and tons of bacteria. These are little coci, the round bacteria. We grow the bacteria with different kinds of media in order to see different things. So we have complex media, differential media, and selective media. Complex media is basically a food source, usually made from yeast, meat, soy, or milk, and like one that we normally use is called nutrient agar, which has yeast in it. Basically, it grows a ton of different things, but we can't really tell what they are. So you can see in this example plate, it's a tan plate. It's just a food source, and it's growing a bunch of pink bacteria, a bunch of pink colonies, but I don't know what they are because they all kind of look the same. Selective media, basically we're trying to figure out what the bacteria are. So the material that is in the agar either enhances the bacteria of certain bacteria or inhibits the growth of others. So basically, if it's going to grow, we want it to really grow, and if it's not going to grow, we want to get rid of it. And it helps to identify certain bacteria. So for example, in this plate, it has a ton of salt in the plate, which I would tell you normally. And so it only is growing skin bacteria. That's what the little S stands for. It's only growing skin bacteria, which is bacteria that can grow in a salty environment. Any other bacteria does not like that salty environment, so it doesn't grow on the plate. So it's selecting for the certain bacteria that can grow in this environment. So compared to complex grows a ton, but we can't tell the difference. Selective only grows a certain kind based on what's in it. And then lastly, differential. It's similar to complex in that it grows a lot of bacteria, but the bacteria differentiates, and so we can tell what they are. So you can see here, we have some white colonies, some yellow, some pink, so it tells us the differences. And even the plate itself is changing color, which we'll talk about in a minute. So differential grows a ton of bacteria, but they look different, so we can tell what they are. So some bacteria plates that we would be growing normally are called mannitol salt agar, or MSA. And let's just break down what's in it. There's mannitol, which is a type of sugar for the bacteria to eat. There's salt. It's 7% salt, which is saltier than most things. It's even saltier than the ocean. And then it has agar in it, which solidifies it into that flat surface. And it also has a pH indicator. It's red because of the pH indicator, which is called phenol red. And when the environment becomes more acidic, it changes from red to yellow, as you can see in this picture. And the reason we see this color change is because some bacteria will break down mannitol. They ferment mannitol, and that changes it to become more acidic and changes to yellow. So let's walk through the specific bacteria we would normally see. We would normally in class have you go and touch a plate with your fingertips and then have you swab. Can't do that right now, but I've actually done it for you, and we'll be doing those experiments in the coming weeks. So this is on page 80 and 81. We have 3 types of bacteria that we're going to be able to see on the MSA plates. We have Staphylococcus aureus, Staphylococcus epidermidis, and Micrococcus luteus. Now you can see that all 3 have the word coccus in it, describing the circular shape that the bacteria form if we were to zoom in really far on a microscope. You may have heard of staph infections. This is Staph aureus is the bacteria that's associated with staph infections. Staphylococcus epidermidis, epidermis, right, your upper layer of your skin. These are all skin bacteria. They all normally are on your skin, and while this may cause a staph infection, you have it on your skin at all times. Doesn't mean that you are more susceptible to staph infections. Aureus means golden, and we'll talk about that in a minute. So when we grow these on a plate, Staphylococcus aureus, the colony is yellow, so it has a little yellow dots all over it. But what we see is the agar changes to that yellow. That's a way to remember. Aureus means golden, the colonies are yellow, and the agar of the plate is yellow. It changes. The pH goes from being more basic to acidic. And why does it change? Because Staphylococcus aureus ferments mannitol. It eats the sugar, which makes it more acidic, which makes it yellow. Staphylococcus epidermidis, the colonies are pink or white. So they are very different from Staph aureus in that the color is different. We can tell the difference between the two. The agar around Staph epidermidis is red. There's no color change, and if there's no color change, that means it does not ferment mannitol. The pH stays the same. Micrococcus luteus has a yellow colony. So if I were to give you a plate, you would see multiple yellow colonies and you'd be like, oh no, I don't know the difference. But luckily, Micrococcus luteus doesn't ferment mannitol, so the agar color doesn't change. So this is the way that we tell them apart. Staph aureus is yellow with the yellow agar because it ferments mannitol. Staph epidermidis and Micrococcus luteus do not ferment mannitol, so their agar around them stays red, whereas epidermidis is pink or white and Micrococcus luteus is yellow. Let's look at that real quick right here. This is yellow, but the agar is red, so these are Micrococcus luteus. Here, the bacteria is pinky white. It's hard to tell because it's more in a streak, but the agar is red, so that is Staphylococcus epidermidis. Whereas here, the colonies are yellow and the plate itself is yellow, so that is Staphylococcus aureus. Sorry, I have to go through those again. So, what type of agar is MSA? Is it complex, differential, or selective? Yes, it is selective in that it only grows bacteria that grows in salty areas. So it's only going to grow skin bacteria most likely because the bacteria on our skin is very salty. It can grow in a salty environment. Any bacteria that can't tolerate that environment will just die and we won't see it growing. But yes, you are also right that it is differential. The bacteria are different colors, so we can tell the differences between them. So MSA is selective and differential. I feel a little bit like Dora the Explorer right there. Okay. Going to transition a little bit to the respiratory system to talk about a different type of bacteria that we'll be looking at. So breaking your respiratory system, you have the upper and the lower respiratory system. You may have heard, oh, I have an upper respiratory infection. I have a lower respiratory infection. A lower respiratory infection is more like pneumonia, whereas an upper respiratory infection is going to be more like a cold. So your upper respiratory system, that's where we're going to be focusing your nose and your throat. So your nose, inside of it, and then your throat, kind of the area, the back of your, where you swallow. Your lower respiratory system is from your larynx down, basically. Your larynx is basically from here, this kind of region right here, down. This blue part is your trachea, which goes to your lungs. You have bronchial tubes, which are these two branches. You can't see because the heart's in the way, and they go to your lungs. And then we have the alveoli over here for gas exchange. We are going to be focusing, though, on the upper respiratory system for this. So the way we look at upper respiratory bacteria is we use blood agar plates. So instead of mannitol salt agar plates, we're using blood agar. Blood agar plates are made of sheep's blood because the bacteria in your upper respiratory system break down the blood cells found in the agar. That's what we're going to grow them today, but that's because they also break it down in your respiratory tract. If you've ever had strep throat and you're like, oh, my throat hurts so bad, it's because the bacteria is breaking down blood cells. It's called hemolysis. Hemo means blood, lysis means to break down. And what we're going to be looking at with blood agar is different strains of Streptococcus bacteria, which is, if you think of strep throat, Streptococcus. Again, you see the descriptor coccus, so it's the round bacteria again. So within your the blood agar plates, we actually have 3 strains of Streptococcus, alpha, beta, and gamma, and we see different levels of hemolysis. So we have alpha, partial hemolysis, beta, total hemolysis, and gamma, no hemolysis. So here we'll look at beta first. Beta is actually the strain associated with strep throat, and it's a total breakdown, and it actually leaves a very clear look. So you could hold this up to the light and you could see straight through it. It's very, very clear. You can even see the person's fingers behind it. That's because the blood cells right there have been totally broken down and used as a food source for the bacteria. Here we have alpha hemolysis. Alpha is a partial hemolysis, so it's partial breakdown, and so it makes this kind of green cloudy haze. So you can kind of see through it, but it's more cloudy, not totally broken down. And then lastly, gamma, there's no hemolysis, so it doesn't look that different. You kind of just see where we streaked the colonies. You can see it up here too. Alpha right here is a green haze. The beta has this clear halo around it, and the gamma just kind of looks like colonies on it. It doesn't really look that different. So, for your homework due April 16th or 17th, enjoy your Easter break. I know it's going to be different, and we know that's tough. I was supposed to host Easter this year as well, and now I'm not. So the ham will stay in my freezer for a little longer. But for your homework, yeah, enjoy the Easter break, but make sure to do your homework for April 16th or 17th. Please read Lab 11 and label the parts of the upper respiratory tract on page 85. Bonus, label all of the parts because the lower respiratory tract is very interesting as well. If you go to page 87, I'm going to do it right now with you. There are a couple circles on the page. What we want you to do is sketch the 3 different shapes of bacteria that you saw, the coccus, the bacillus, and the spirillum. So draw another circle and draw all 3 of the shapes. Oh. Then you're going to sketch and label the 3 different types of Streptococcus bacteria. And I would use color. So you can draw, and I'm sorry, I misspoke. So for this one, in the first circle, go ahead and draw all 3 shapes in the circle. You don't have to draw 3 different circles. And then in the second circle, I want you to draw the Streptococcus bacteria colonies. So the alpha, the beta, the gamma strains. Show me what that would look like and use color. So do it. I would do it similar to this, so you remember what the shapes are and what the colors are. We will be posting the Science News article on strep throat, and you'll be answering the 6 questions, on page 88 and 89. It's like our normal Popular Press articles we did before the break, just on strep throat instead. It's due on Canvas. For my class, it's April 17th, but please check with your class to see when it is due. 6 points again, and you can submit on Canvas either as a text box or a Word doc, up to you. And then the last thing is to complete the Lab 11 lab report. It's also, I think, going to be called Lab 11 worksheet. Due on Canvas, again, 16th or 17th. It's 20 points, open note. Get all the points here that you can. If you have any questions, please let us know. We'd love to help you. We hope you have a good Easter break, and let us know if you need anything. Stay safe out there.